Our next presentation is on the Kansas City campus. Student Dr. Aziza is originally from Alaska and attended DePauw University for her undergraduate career. They majored in biology and after graduating will be applying to PA programs in hopes of becoming a physician's assistant specializing in gynecology. So the floor is yours.
Hi. Hello, everyone. My name is Aziza. I'm going to be talking about the research I've been doing over the last 2 years, titled Investigating the Role scaRNA2 Plays in Modifying mRNA Splicing in Relation to Congenital Heart Defects. Okay, first I wanted to talk about congenital heart defects and why these are important to me. These make up 25% of all birth defects, and they're the leading cause of death within infants. I'm interested in a specific congenital heart defect called Tetralogy of Flow, which represents 5 to 7% of CHDs. This is a combination of 4 different heart lesions that are present in this diagram over here, and they affect blood oxygenation and the flow of blood throughout the heart and to the lungs. And surgical intervention is required within the first year of life. The genetic causes underlying this are still unknown, so that's where I'm hoping to come in. So in the past, our team has looked at heart development and has been able to tie it to alternative splicing. We found that about 50% of genes associated with heart development were alternatively spliced within the right ventricle of infants with the heart condition Tetralogy of Flow. And we've also seen that scaRNA levels are linked to spliceosomal maturation and function during heart development. And the scaRNA that I'm interested in, which is scaRNA2, was seen to be reduced by 40% within samples taken from babies, the right ventricle of babies with Tetralogy of Flow. So I wanted to talk about the spliceosome and what exactly it does. This is a large, complex molecular machine. It's located in the nucleus, and its job is to perform mRNA splicing. It's made up of subunits called spliceosomal RNAs, or snRNAs, as well as some proteins. And I'm interested specifically in snRNA-U2, which is over here. This is an important component of the spliceosome, and it's where my scaRNA2, is focused. The process that the spliceosome performs is something called splicing, which is the removal of non-coding regions of mRNA called introns, and the joining up of the coding regions, which are exons, in transcribed pre-mRNA. Another important function of the spliceosome is alternative splicing. This allows one gene to produce multiple mRNA isoforms, causing different protein isoforms to be produced and allowing for protein diversity. I have some quick examples of how exactly alternative splicing works. These are very simplified. It's a little bit more complicated than this. But basically, if you've got your 3 exons, they might come together in 1 and then 2, whereas in alternative splicing, they would come together in any other order other than 1 and then 2. So anything that's alternative. So I'm interested in snoRNAs. These are small nucleolar RNAs. These are a class of RNA molecules that guide post-transcriptional modification of both rRNA and snRNA. And in my case, I'm interested in snRNA because these are the components of the spliceosome. I am specifically interested in a subfamily of snoRNAs called scaRNAs. These are essential for biochemical modifications of snRNAs, and in my case specifically, they perform methylation, which allows for changes in RNA atom arrangement. And I am specifically focusing on scaRNA2, which methylates 3 different sites. And you can see those 3 sites identified here with these 2 diagrams. So now we come to my big picture, which is I'm interested in why is scaRNA2 involved in heart development? So the way that I went about trying to understand scaRNA2's involvement was by first growing and transfecting a cell line, a human cell line, with a scaRNA2 knockdown. I then was able to measure the level of knockdown by assessing the change in methylation level. So I was looking at a decrease in the actual functionality of scaRNA2. And from this, I then sent RNA samples off for RNA-seq data to assess gene pathways that were affected by my scaRNA2 knockdown, as well as changes to alternative splicing. From these methods, I got a couple of significant results. I found that scaRNA2 was successfully knocked down by about 35% on average among 5 different samples. And then I went and quantified the methylation, and I saw that there was about a 30% decrease in methylation on average among a couple of samples. And from here, I then performed RNA sequencing, which allowed me to assess alternative splicing. And from this, I found different protein isoforms that were produced between my knockdown and my control samples. And I saw many pathways that are involved in heart development were affected by my scaRNA2 knockdown. I was also able to identify genes of interest, and this was a pretty massive list, but I'm only going to be talking about 2 here today, which are CSTF1 and MYL3. Oh, and the reason that these genes are important to me is because they're involved in development, specifically of the heart, in MYL3's case. So here's some of my actual data. You can see on the left, I've got my scaRNA2 knockdowns all put into just 2 simple bars. Compared to my control, they were decreased by 35%. So this is an important number to me because in actual heart tissue of babies with Tetralogy of Flow, we saw that scaRNA2 was decreased by about 40%, and I had a decrease in 35%, which is a little bit different, but I feel like it allows me to say that these samples are similar to this heart tissue. From here, I then assessed the methylation, and I saw that there was a decrease of about 30% in scaRNA2's actual functionality. So then all of this allowed me to send my samples off for RNA sequencing, and I got back my list of genes that had changes in functioning. So MYL3 was one of these genes that I found that was changed during my knockdown. And you can see up at the top here, there are 9 exons involved in MYL3. During transcription, these are going to be spliced together like this. And when all of these exons are present, so all 9 of these are present, this is the protein that is predicted to be formed. But in my samples, I saw 2 different splice isoforms. So I saw this one here in the center, which is missing exon 1 as well as exon 8 and 9. And this isoform was actually increased within my knockdown samples. And you can see just from looking at it that this protein is predicted to look pretty different from the one with all of the exons present. I also saw a different isoform that stopped prematurely through exon 7. And this produced a slightly different protein as well. So then I got similar data for the gene P10, which is also involved in development. And you can see that this also has 9 exons. They're transcribed together. And then here's the normal protein isoform that we predicted. And you can see that my protein isoform is actually a lot smaller than the one predicted with all exons present. It only actually has 3 exons within it. So from this, the conclusions that I was able to draw were that scaRNA2 level is important for mRNA splicing, and it affects processing of many transcripts important within development. It causes alternative splicing, and ultimately it changes the predicted forms of proteins. And we found that when scaRNA2 is knocked down, we saw altered gene isoforms involved in pathways important in heart development. And this diagram is a little bit difficult to read, but basically the genes that we looked at before are involved within these pathways present here. And you can see that these pathways range from all areas of the cell, they're within the plasma membrane all the way to the nucleus. And these are the connections that, or these are what those pathways are associated with. So you can see that a lot of these functionalities are involved with the heart. So a lot of the pathways that I saw had involvement within the heart and heart failure. So I wanted to say thank you to my lab group and all of the other students involved, as well as my board members. Here are my sources.
Hi, thank you. Aziza, that was a great talk, great data. I just have a very simple question. I've always asked Dr. Biddle about this. So the thinking is that the scaRNA2 changes that we see in Tetralogy of Flow, it is causative and not simply responsive to the disease state?
I would say that it is causative. Well, I'm not 100% sure. That is a very good question. We would have to look at heart tissue from before the baby has actually developed, and we haven't had access to that yet. So I'm not 100% sure.
Just jump in there. Aziza will be defending in about a month, so come and learn more about this. Knowing whether it's causative or a consequence is a really hard thing to do. Our evidence that it is causative is we've done it in a couple of animal models. So when we interfere with the expression of these non-coding RNAs, it impacts heart development as well as alternative splicing. So that's pretty good evidence that it is involved in the development of the heart, but I wouldn't say we have yet proved it. And that's basically what Aziza said.
Hi,
Okay,
from
couple more
Joplin.
questions.
Just a question. How did you prevent the RNA from like denaturing while you were doing your research?
So I kept it on ice most of the time that I was working with it, and when I wasn't using it, I stored it at -80. It was only out on the bench for short periods of time. And we actually converted it to double-stranded DNA for qPCR, so it was in a more stable configuration.
Perfect, thank you.
Thanks.
Clearly a question from someone who's worked with RNA, because those of you who have know how unstable it is. Okay, I think Dr. Zaidi
Yes.
and then Dr. Sarsour.
Excellent presentation, Aziza. My question has to do with you mentioned that the knockdown efficiency was 35%, but you had like 5 cell lines or 5 separate samples. So my question is like, did you have a sample which had like a 90% knockdown and another with 10, and the average was 35, or how do they compare?
So the average was 35, and I had one sample that was significantly higher than the rest as well, but the rest of them were like more in that 30%, 35% range. I also had some samples that didn't show a knockdown at all, so it really depends.
So in your RNA-seq data, this is kind of the average of all of the 5.
Yes.
You pulled those samples.
It is, yeah.
And then my next question has to do with you mentioned that the original protein would be like a large size, I forget the size of the protein, but then the ones with the fewer exons would be a smaller protein. So like in reality, is there a situation where you see the right size protein and a truncated protein? Is there like a mixture that appears in these babies, or one form is predominant?
That's a great question. I am not sure about how predominant any of them are. All I know is that the larger one that you see is the one that's present if all exons are present. I don't know exactly which one is more common. And I'm sure that there's going to be different levels throughout development. So that would be something that I would need to get into in my thesis.
And does anybody know if the truncated version of the protein is functional?
So we know that it's protein coding in at least 3 of these cases, but this one that you see here, it's not even protein coding. So I would guess that one is not functional, but I'm not sure about the others.
Thank you.
Can I jump in there again just a second? There's a dramatically increasing interest in alternative splicing. And last week at the BioNexus conference, we had a guest speaker from Nebraska who, if you were there, you saw that one of the things he is helping his colleagues do is identify functional domains that may be important in alternatively spliced messenger RNAs in the resultant protein isoforms. So it's a good question. We don't know the answer yet, but it certainly is critical to understanding the impact of alternative splicing. I might also add, your first question, Dr. Zaidi, was a really good one. Come back next month. We know that's a good question, and Aziza will have some data about that next month.
Way to go, sounds like a party.
Question
Dr.
in
Sarsour.
Joplin. Oh, I'm sorry, go ahead.
Dr. Sarsour, go ahead, and then we'll come back to Dr. Staudinger.
Okay, so my question is, when you knock down, you enrich the other splicing form, right?
Sorry, could you repeat that?
When you knock down the RNA, you enrich for the other splicing forms, or what? Is that what happens?
I'm sorry, could you repeat that?
Do you enrich for the other? So is the other splicing form are enriched? That's what happens?
I'm sorry,
Especially
I
for
don't think I
P10,
understand.
because this is really important. I'm really interested in P10.
Let me see if I can reinterpret your question for Aziza. So when you knock down scaRNA2, the alternative forms, are they enriched or depleted? Is that correct?
Yes,
Oh, so the other scaRNAs, you mean?
No.
No?
Is it the other scaRNAs?
the gene that been spliced is the other
Oh,
splicing
so-
forms.
Aziza, just look at what you've put up here. So for this particular gene, P10205 is going up,
Yes.
P10202 is going down. So I guess that's the answer.
Okay. I'm so sorry. So you were just curious about increasing versus decreasing levels of these isoforms? Yeah, okay, I'm so sorry. Yes, I do know that 205 is increased and 202 would be decreased within these knockdown samples.
And we, like examine these sequences, like do you have primers for those?
I do have primers for these, yes.
Okay,
Yeah.
because I'm really interested in P10 and.
Oh, I
if-
don't
if
have
that's-
primers for the.
If that's change also happens in cancer cells, because
Mhm.
P10 and cancer cells is still normal, but there is something in its pathways happening. Maybe that's the splicing.
Gotcha. So I don't have primers for any of these genes. These genes I just found through RNA sequencing data. So I didn't do any actual wet lab research with these.
Okay. Dr. Stottinger, did you have a question in Joplin?
Not so much a question, but I'd like to share something with the audience. And in the Stottinger lab, we work a lot with RNA. But then there's Dr. Rogers, who is a protein biochemist, and we're collecting all kinds of sayings and to make T-shirts on them. And one of my favorite sayings of his is, RNA can smell fear, which is true.
For anybody who's worked with RNA, yes. It
Absolutely.
smells fear and then hides, it disappears. Okay, any final questions? Okay, then thank you, Aziza.
Thank you so much.